2020 is a special year. It's a chance for humans to take hold and rethink our living styles.
We used to live with comfort, the commodity seems infinite, while the crisis reveals that the truth runs opposite way, resources are finite.
The concept of fair distribution of resources could only be achieved in real infinite ways. The idea is simple: sunlight. We see its potential in synthetic biology.
The notion of hybrid car can be a perfect analogy of the energy conversion system. What if one day we could directly use sunlight as part of the energy source? We're interested in finding the integration of such a concept into food-eating organisms. From a classical start, of course, E. coli.
NCTU Formosa proudly presents this unique strain of E. coli E-hybrid as our fundamental strain for creative applications. Before introducing this E-hybrid, I want to talk about a special protein that is crucial to our project, Glioblastoma rhodopsin, also known as GR. GR originates from primitive cyanobacteria, Glioblastoma violaceus. It is a 7-helix membrane protein located in the inner membrane. Acting as a light-driven proton pump, GR can transfer protons from cytoplasmic region to the periplasmic region following the light absorption. That is, it establishes the proton motive force to push ATP synthesis, transforming solar energy into universal energy currency, ATP. The reason that GR has a function with light is its specific chromophore, or transretinal. It changes its conformation when induced by light, resulting in a series of protonated and deprotonated reactions on the several amino acids in GR, and causing the transfer of protons. If we design an E. coli with this special chromophore and GR protein, E. coli can use light as an alternate way to produce energy, meaning it could be a second engine apart from respiration, decreasing metabolic stress and thus enhancing efficiency. This is our BioBrick design. This composite part is regulated by a T7 promoter. A polyhistidine tag is used for purification. In order to assess expression, we use a linker sequence in junction with GFP. Besides, in our experiment, we'll use ultransretinal as GR's chromophore. We produce beta-carotene, the precursor of ultransretinal by ourselves. We use the BioBrick CRTEBIY, which is from iGymKit, to produce beta-carotene. Finally, we use RFP regulated by leg-guide promoter to prove our concept, which is the better production in our GR expression system. Now that you're familiar with our BioBrick, here's Julie to explain our experiment design.
The experiment design consists of 3 parts. First, we build up the gene construct of Glioblastoma rhodopsin and verified its expression. And then, we tested the function of Glioblastoma rhodopsin in direct photocurrent measurement and phototropic growth measurement to evaluate its effect on living E. coli. And finally, we focus on whether E-hybrid could improve protein expression. For the first part, this is the cloning result of our project. Glioblastoma rhodopsin and its chromophore we use, beta-carotene, all falls in the correct ranges according to their respective base pairs. We successfully cloned Glioblastoma rhodopsin in expression vector PET32A and produced beta-carotene and extracted it with DMSO. It is commonly acknowledged that membrane protein is a difficult target for expression. So we choose Demo21, which has a tunable T7 expression system for the better harness of the protein expression rate for the proper folding into E. coli's inner membrane. The proper folding of Glioblastoma rhodopsin can be visualized by green fluorescence. We tried various growing conditions in low temperature and varying air remnants. Much to our surprise, GR shows the strongest GLP signal with IPTG induction without remnant inhibition, meaning that GR can be easily expressed by E. coli. After the successful expression, we next evaluate the function of Glioblastoma rhodopsin in living E. coli in 2 ways. First, we measure the proton pumping amount of Glioblastoma rhodopsin by detecting the photocurrent under intervals of light and dark environment. GR expression E. coli show a significant increase in photocurrent under light excitation, thus proving its proton pumping activity. The second way to evaluate GR expression E. coli is to observe its growing behavior with the expression of proton pump, whether it will give positive effect on its metabolism and be the alternative of the electron transport chain. So we did several phototropic growth measurements and found them growing faster than the vector control ones. To further investigate the role of glial beta-reductase in chemiosmotic process, we inhibited respiratory electron transport chain by adding cytochrome C inhibitor, sodium azide, and under the growing environment with the addition of sodium azide. GR expression E. coli survived the pressure, which indicates that glial beta-reductase do serve as an alternative for respiratory electron transport chain. Finally, we want to know whether the target protein expression will increase with our GR expression system, which makes E hybrid serve as a fundamental strength for bioengineering success, so we incorporated the RFP expression. The highlight is that when we culture E hybrid with RFP, we will get the inspiring fact. Let's look at the graphs. The expression of RFP is better than E. coli without GR expression system. From the experiment, we proved that E hybrid can incorporate with different target proteins for various applications. To learn more about how we further investigate the function of glial beta-reductase, let's look at our modeling results.
The ultimate goal of the model is to provide users with useful information on optimal growing conditions of E. hybrid, driving the growth of GR expression E. coli with model simulation. So, we built our growth and consumption model. First, we built our E. coli growth curve pattern and the corresponding glucose consumption simulation, which predict the growth curve with various glucose uptake rate of the wild-type E. coli and the E. hybrid, as the graph shows. We expected that the glucose uptake rate should be influenced by additional ATP. The figure presents the fact that the higher glucose uptake rate, the faster the growth rate is. Hence, we propose a hypothesis that E. hybrid will grow faster, and the experimental results were also in line with our initial hypothesis. The following figures are the prediction of the growth model, and we tested it by independent experimental data. Moreover, the corresponding glucose consumption is as follows. The glucose consumption and its exhaustion point is highly correlated with the growing pattern, and we through the prediction we expected that E. hybrid might consume the glucose faster and thus reach the stationary phase earlier, with earlier glucose exhaustion. Based on the growth and the consumption model, we were able to further analyze the growing pattern of E. hybrid and gain some useful information for optimal growth with different needs. Let's consider the optimal concentration of glucose. The glucose mainly determines the overall lifespan of the bacteria. Through the simulation, we interestingly found a glucose threshold for expanding lifespan. We expected that the E. coli would decline when the glucose is exhausted. We can see that E. hybrid needs at least 33 millimolar glucose to efficiently delay the decline time from the slope increase after 33 millimolar. After analyzing the decline timing of E. hybrid, we can state about optimal glucose amounts for different growths. Another important part of optimal growth is to seek for the best refreshing strategy for continuous protein expression for maximizing yield and minimizing waste. So we made predictions on the timing of refresh with different growths. If we intend to let E. hybrid constant survive with its function for more than 24 hours, we recommend the glucose concentration be 40 millimolar and the refresh ratio of 1 to 2 with fresh medium. In other cases, if we hope to achieve efficient protein yield in a short time, an opposite ratio of 2 to 1 is recommended. Eventually, the optimization growth model is integrated with hardware. We verify this model to be useful for the optimization growth in the device with successful prediction.
innovative engineered E. coli shouldn't be confined in the bioreactors in labs, as our E-hybrid can thrive with the help of sunlight. In this light, we design a culturing device allowing for efficient protein expression. We name it the Smart Solar Incubation System, abbreviated SSIS. In order to take our device out from the lab, we first simplify the function of the bioreactors and retain some critical parts. And we design additional elements for biosafety and remote control. It mainly consists of 4 parts: a shell for protection, a mini incubator, a solar stove for sterilization, and a storage unit. With this demonstration, we want to tell users that it's possible to build a natural incubated tank in the wild without the requirements of continuously putting nutrients. Let's take a look. The shell was made of transparent acrylic attached with a layer of anti-UV film, so sunlight could directly beam on the incubator and minimize the possibility of E-hybrid mutation. The body of the device was made of wood for basic protection against the rainfall, and at the same time, we expect a balance of temperature and humidity between inside and outside to maintain its culturing condition. Considering the problem of biosafety, we established a solar stove to sterilize the product using solar heat. The whole hardware devices are also powered by sunlight, with solar panels attached around the acrylic outside the hardware. The remote control can be achieved by using IoT talk system. Sensors of weather and culturing information such as OD600 value, temperature, and light intensity could be uploaded to the online IoT talk, which provides information for users. Last but not least, the optimization growth model is also integrated into the software for users to maximize the production. NCTU Formosa envisions the future with E-hybrid in daily usage with SSIS. For the implementation, we must go out of the lab, reach out for people, and know the demand of the public. So, human practices is a must.
To put E-hybrid to real world, we should often listen to public opinions. To promote E-hybrid, we should seize every opportunity for science communication. And to fulfill E-hybrid, we need help from our iDream team. So here comes our human practice. Our human practice consists of 3 parts: integrated human practice, science communication, and collaboration. Our integrated human practice includes 3 target groups: professors, the public, and enterprises. First, we consulted Professor Gao about the way of optical measurement to measure beta-carotene accurately. Next, we held conference and made a carbon resource consumption survey to collect opinions from the public. Last, we visit Graphene Bio and T.Bio. T.Bio is an enterprise of synthetic biology. They have inspiring talk with us in E-hybrid application of medicine manufacturing. Finally, we have done several SDG in Hsinchu. Integrating all this feedback and advice, we benefit from every phase and gradually share the project. For science communication, being able to communicate with other groups is critical, and so there are so many hindrances this year.
We have held the NCTU and NTHU conference, high school workshop, NCTU presentation, and participate in Taiwan iDream conference and other online communication, social network, and online meetup. In addition, we record content about our project and iDream with GoTo and Play, which is a radio channel in NCTU.
After doing this science communication, we do stretch our ideas out and also take back some valuable advice.
Last but not least, we collaborated with all iDream teams in different aspects and built a partnership with NTHU Taiwan. Especially, we have collaborated with NTHU Taiwan in many aspects, including dry lab and wet lab. Both of us witnessed the development of our projects.
Hybrid presents itself as a simple living paradigm for retaining sustainability and for pursuit of efficiency simultaneously in modern ages. Simple as E-hybrid's design is, it has the potential to benefit countless fields and industries. It can improve production efficiency of much needed medicine, like antibiotics, novel protein drugs. It can pave new avenues for effective bioremediation by mass-producing protein that binds to heavy metals and other types of pollution. It can help us finally achieve green agriculture by replacing chemical insecticide and fungicide with all natural peptides that target pests while preserving soil and natural integrities. Above all, eHybrid can harness the incredible versatility of synthetic biology to enrich productivity and provide critical resources to those that need them the most. The events of this past year serve as a dark reminder of obstacles we have yet to overcome. However, we firmly believe that our project serves as an initial step toward ensuring that people of all walks of life are provided for, while unburdening our planet's valuable and finite resources. As our school motto goes, "Be the light of the world," we sincerely hope that eHybrid can be the shining light at the end of the tunnel that leads humanity into a brighter and better future for all. Thanks to our team's members, instructors, advisors, and sponsors to help us make our project. This was the presentation of NCTU Formosa. Thank you for listening.